Today, I want to talk to you guys about war. All of us have heard of war. We've seen war on the news, and we've studied it in school. And some of us have even been firsthand witnesses to war. I am one of those people, but not to the kind of war that you're thinking of. Growing up, my dad had multiple sclerosis, and as a result, he was very prone to contracting urinary tract infections, and so he was often on antibiotics to resolve these infections. One time when I was 13, or sometime within that frame, my dad had contracted a urinary tract infection, and as a result, he was on antibiotics, and so I didn't really think much of it. But later that night, I found myself in the emergency room with my dad. When I walked in, he was connected to every tube imaginable. He was unconscious, unable to talk, unable to breathe on his own, and he was truly in a battle for his life. I felt like I was looking at two completely different people. I didn't understand how my dad could go from seemingly healthy that morning to fighting for his life just later that night. Upon talking to the doctor, he mentioned that my dad had something known as sepsis. Sepsis is a life-threatening complication due to an overreaction of the immune system to a bacterial infection in the blood. 1 in 3 patients who die in a hospital die of sepsis in the United States. There are 48.9 million cases per year and 11 million sepsis-related deaths worldwide. Now, to further investigate the mechanism that results in sepsis, it's important for us to first take a look at the makeup of a bacterial cell. So, gram-negative bacterial cells have an outer cell membrane, which contain a lipopolysaccharide layer. Lipid A is a further component of this lipopolysaccharide layer and is regarded as an endotoxin. So, when present in the bloodstream, our immune systems, or cells that work for the immune system, detect lipid A, and in response, release cytokines. When these cytokines are released, other immune cells are signaled to migrate to the area of infection, to clear it up, or to mitigate damage, and typically work just to resolve the infection. But in the case of sepsis, there are too many cytokines released and too much of a reaction from the immune system, and that can result in organ damage and organ failure. So it's a little bit like having too much of a good thing leads to a bad thing, where you have just too much of an immune response that would typically work to clear up the infection, but in this case results in more damage. Sepsis is further complicated by antibiotics, which, as I mentioned, are a routine treatment for bacterial infections. So when treated with antibiotics, bacterial cells lyse or explode and release their contents into the environment that they're in, which in this case is the bloodstream. When this happens, lipid A is released and causes the immune system to overreact, which leads to organ failure or organ damage and can result in long-term disabilities. So now our question becomes, how can we work to, 1, resolve bacterial infections, and 2, prevent the immune system's overreaction to lipid A? And this leads to the 3 objectives that I want to talk to you guys about today. 1, implementing a new way to treat bacterial infections. 2, finding a way to prevent the immune system's response to lipid A. And 3, once we have those 2 things figured out, finding a feasible way to produce whatever product that will work for this in lab. So, in regards to our first objective, through literature searches, we were able to identify phage therapy as a potential strategy for resolving bacterial infections. So phages are viruses that are specific to bacteria and are very efficient delivery machines. Historically speaking, phage therapy has been used as a method to treat antibiotic resistance in bacterial cells. And when treated with phage, bacterial cells lyse open and, again, just like with antibiotics, release their contents into the environment they're in. So now we have another method that we've found that can be used to resolve bacterial infections, but remember that the problem we're trying to prevent is the immune system's overresponse to lipid A. So the way that we can do this is with anti-lipid A proteins. Here is a list of candidate anti-lipid A proteins that we, the Ohio State IGEM team, constructed that we could potentially use for treating sepsis and preventing the immune system's overreaction in sepsis. And whether it's binding to lipid A or modifying it in some way, the idea is that in the presence of the anti-lipid A proteins, the proteins will bind to lipid A and therefore prevent the immune system's over-response to lipid A, which will prevent the effects that sepsis can have. So now this takes care of objectives 1 and 2. We have phage therapy as a way to resolve bacterial infections, and we have anti-lipid A proteins as a way to prevent the immune system's over-response to lipid A. So now we have to figure out a feasible way to construct this in lab so that we can eventually use it in a clinical setting. In order to figure this out, we first have to look at the life cycle of a phage. So this is just kind of a general overview of a phage's life cycle. So first, the phage attaches to bacteria with specific receptors. It then injects its DNA into the host cell and uses that host cell machinery to replicate, similar to how human viruses do that in our bodies. And then once the phages have replicated so much, the bacterial cell won't be able to hold them anymore, and as a result, will lyse open and release them into the environment. Now remember, at this stage, we have incorporated our anti-lipid A proteins. So the idea is that we'll have the DNA sequences for our anti-lipid A proteins that will be cloned into the phage. And then when the phage injects or attaches to the bacteria, it will inject its DNA as well as the DNA sequences for the anti-lipid A proteins, which will be transcribed and translated within the bacterial cell. And then once the bacterial cell lyses, it will release the proteins along with the lipid A, and the anti-lipid A proteins can bind to the lipid A to prevent the immune system's over-response. However, now we run into another problem, which is how can we grow our phages when the genes that we're inserting might be lethal? Recall that lipid A is a component of the lipopolysaccharide layer, which is a component of the outer cell membrane of gram-negative bacterial cells. If you're putting anti-lipid A proteins into the bacterial cell to be transcribed and translated, then this can cause problems with bacterial cell growth or function and may result in us not being able to actually grow our phage with the bacterial cells in lab. And so the way that we can resolve this is with a double promoter system with an anti-lipid A gene downstream of our promoters. So the first thing I want to note with this slide is that the purpose of having 2 promoters is to maximize protein output, because eventually when we are operating in vivo, we will want to have the maximal amount of anti-lipid A proteins so that we can bind to as much lipid A as possible to prevent the immune system's response to the lipid A. Now, the star of the show here is the P4B promoter, which is interestingly a promoter taken from Salmonella that we took the plasmid for and put it in our circuit, which will then be cloned into our phage, and then will be used to transcribe and translate the anti-lipid A proteins in a bacterial cell. In the presence of FRA-R, which is the repressor for the P4B promoter, we can control gene expression because the FRA-R repressor will bind to the P4B promoter and allow us to decrease the amount of anti-lipid A protein being produced. And this should allow us to be able to grow our phage in lab and to prevent the disruption caused by the disruption to bacterial cell growth and function from the anti-lipid A proteins. So now that we have our design, we have to figure out if it actually works the way we expect it to. And we have done this with luminescence assays. So as you can see in this figure, we see that in the presence of FRA-R, that we see a decrease in gene expression measured using a GFP or green fluorescent protein reporter. And this tells us and confirms our expectations that we would see a decrease in the amount of gene expression, which when we put our anti-lipid A genes in place of the GFP, then we will be able to see a decrease in the amount of total anti-lipid A protein output. So now that we've confirmed that our genetic circuit or double promoter system works, we have to determine how well our anti-lipid A candidate proteins work to actually reduce the amount of lipid A so that we can reduce the immune response. And we've done this with a LAL assay. So as you can see here, to the far right is our control, and then we also have other conditions in bacterial cell populations with different anti-lipid A proteins and promoter combinations. The LAO assay allows us to measure endotoxin unit concentrations present in a given cell population. And so as a result, we can see that relative to our control, the majority of our anti-lipid A proteins result in a decrease in the total amount of endotoxins present, which will allow us to decrease the immune response that occurs when lipid A is present. So now that we have this preliminary data, our next question is, well, what are our next steps? So our next steps will be to clone proteins into our phage. We'll then test this with a HEC blue assay, which will allow us to calculate endotoxin concentrations so we can determine how much we're actually decreasing the endotoxin levels by. And then we'll do murine or in vivo model studies so that we can simulate an environment that's more similar to the environment that our phage will eventually be operating in the human body. So to summarize, we said that a bacterial infection can lead to sepsis through the immune system overreacting to the presence of lipid A. The Ohio State IGEM team has designed a phage to produce an enzyme that eliminates, binds to, or modifies lipid A in some way to reduce the immune system's over-response to lipid A and to prevent the effects caused by sepsis. So to close out, I wanted to show this picture of me with my dad about 2 to 3 months into his battle with sepsis. While he was able to make it through his hospital stay and even through rehab at the nursing home and to come home eventually, he was unable to survive the long-term effects that sepsis had on his body and passed away in 2018. I hope that in the future we can utilize phage therapy as an effective method to prevent or mitigate the long-term effects caused by sepsis. Thank you. Thank you.